I'll do a few small presentations on the genetics and psychiatry. It's going to be a series of several slide groups. So I'll start with the human genome and talk a little bit about, in general, about the DNA, the chromatin, the chromosomes, genes, and then about the DNA sequencing. So the human genome contains about 3 billion nucleotides, or also called base pairs. These 3 nucleotides or base pairs in the DNA codes for one amino acid. In this slide, we see the groups of the nucleotides: guanine, cytosine, adenine, and thymine. This whole human genome consists of about 2 meters of DNA wrapped around histone proteins. Histone proteins are round, and the long string of the DNA is wrapped around it. And in order to be transcribed, the DNA has to be unwrapped. There are 2 coils of DNA wrapped around a histone protein, and this is called a nucleosome. Many nucleosomes form a chromatin loop, so you can see in this slide actually chromatin loops as being part of the chromosomes, and then the DNA double helix being wrapped around the histones. So a chromatin is a loop of DNA that contains about 100,000 nucleotide pairs or base pairs. Chromosomes are made of chromatin, and the humans have 46 chromosomes or 23 pairs, which are half of them are inherited from the mother and half of them from the father. In fact, there is something called recombination, and the DNA, the entire DNA that we have, is divided into 46 chromosomes. Now, what are genes, and where are they located? Now, genes are functional units. That means that genes cannot be microscopically visible. It's not something that I can look in the microscope and see, like I see the chromosomes or the chromatins. Geneticists divided the DNA into groups of nucleotides that code for one single protein. So a group of nucleotides on the DNA that code for one protein is called a gene. And the genes have a variable number of nucleotides. Genes contain from about 1,000 to more than 1.5 million nucleotides or base pairs, depending on how large the protein that they are coding for are. So here in this slide there's a protein called titin, and it's a very large protein that is contained in the muscles and the heart muscle, and contains about 27,000 amino acids. And remember, there's 3 nucleotides that in the DNA that code for one amino acid. A Human Genome Project was completed in 2003, and it was established that the human genome contains about 20,000 to 25,000 genes, which is about in the middle of the other animals and plants. So, for instance, the fruit fly contains about 13,600 genes, while the rice, for instance, contains more than the human genome, about 45,000 genes. The genome is made of coding, non-coding, and regulatory parts. Only 1.1% to 1.4% of the genome sequences code for proteins. So in this slide we see the exons, which are the coding regions, and introns are the non-coding regions. So in addition to the coding and non-coding regions, we also have regulatory regions. We have just started to learn some more details about the introns. In the beginning, when the DNA was when the genes were discovered and tested, and we did not know much about the introns. So at one point in time, this part of the DNA was called junk DNA because it's a part of the DNA that we didn't seem to know what it was doing. So these non-coding regions, or the introns, are initially copied into RNA. So the first copies of the RNA contain both exons and introns, but the final copy contains only exons, and the introns are cut out, like you can see in this slide. Now, regulatory regions of the DNA, about 10% of the genes in the human genome code for DNA binding proteins or regulatory proteins. These are parts of the genome that do not form proteins. They regulate when the proteins are going to be formed. Some of these proteins recognize and attach to specific portions of the DNA to activate the gene expression. They can be transcription factors, they can be nuclease, polymerase, and so forth. Now, variations in the human genome are called mutations, but a genetic variant is a permanent change in the DNA sequence that make up a gene. And most variants are harmless or have no effects at all. However, other variants can have harmful effects and lead to diseases. The single gene mutation, some genetic variations affect only one single gene. Single gene mutations are responsible for many rare diseases, such as neurological diseases, such as Huntington's disease. For instance, in Huntington's disease, you have a repetition of a triplet more than 40 times, which leads to a repetition in the Huntingtin protein more than 40 times that leads to Huntington's disease. In the Huntingtin gene, triplet repeats of about 20 to 30 times are normal and cause no symptoms. Now, in people with Huntington's disease, the number of repeats reaches 40 or more. The mutation creates abnormally shaped proteins that are toxic to the neuron. And as the cells start to die, the symptoms of Huntington disease appear. And they consist of uncontrollable movements, writhing movements of the legs and arms, loss of muscle coordination, change in personality and thinking, and also psychosis. In Lewy body dementia and Parkinson's disease are linked to copies of the SNCA gene. So in this condition, we have more copies of this SNCA gene, and this gene encodes for a protein called alpha-synuclein. And the excess of alpha-synuclein accumulates inside the brain cells, jamming the cell machinery. So this is what we see in Lewy body dementia, in which we see mostly symptoms of Parkinson's as well as psychosis, mostly visual hallucinations. Now, some diseases are multifactorial, meaning that they do not depend on one single gene. They show familial clustering, but they do not conform to any recognized pattern of single gene inheritance. They are termed multifactorial or complex diseases. Now, these disorders depend on many genes and the environment. Next, I'll be talking about how do we find these abnormal genes. How do geneticists look for the genes? Where do they find them? So basically, they search for connections between genes and diseases by performing three kinds of studies. The first kind is the linkage studies, and they can use DNA or RNA, and linkage studies is done in families in which one disease is prevalent. For instance, one of the schizophrenia genes was called DISC1, was discovered through a linkage study in a family in Scotland. Another kind of study is genome-wide association studies. They usually use DNA and gene expression profiling, which uses RNA. So basically in gene expression profiling, you are looking for the protein that is already made from the genes. So the protein was already transcribed. When the DNA or RNA portion is presumed to contain a susceptibility locus for a disease, that area is read nucleotide by nucleotide. So this is called DNA or RNA sequencing. Now, nowadays, DNA and RNA sequencing is done by microarray. But in the past, it was laboratory bench work. You would have to do gene by gene. But today, regardless of which study it's used, whether you have a linkage study or genome-wide association study, the end result is the DNA or the RNA sequencing. So you need to find out the sequence of those genes in the string of the RNA or DNA. And this device, the microarray, is employed for that. It consists of a small chip or a gene chip that's coated with row after row of DNA or RNA that is used in sequencing. Now, next generation sequencing is even more complex. It's a new set of techniques called the NextGeneration Sequencing, which emerged as an efficient and cost-effective way to get a detailed readout of the genome. For example, this Lindsay approach that it's here in the slide has the potential to sequence DNA at the speed of hundreds of thousands of base pairs every second. These genetic studies will be used for mostly in preventive, as a preventative strategy. In laboratory, genome-wide association studies and gene expression profiling studies are leading to new insights into the possibilities for disease prevention. When geneticists identify a gene or gene regulatory pathway associated with a disease, they uncover potential new targets for therapy. Understanding the relationship between genes and complex diseases, it's also expected to play a role in personalized medicine. One day, microarray-based genome scanning could become a routine way to estimate a person's genetic risk of developing a disease like stroke, Alzheimer's disease, Parkinson's disease, schizophrenia, or certain brain cancers. Also, researchers hope to develop customized drug cocktails that are matched to a person's unique genetic profile. And researchers believe that these customized drugs will be much less likely to cause side effects than the medications that we have now. Thank you.